Welcome to Brain Ponderings, your window into the world of brain research with unfiltered conversations with experts in various aspects of brain science. And today, my guest is Professor Magdalena Götz. She's in Munich and she has various titles there, I think. She's professor and head of the Institute of Stem Cell Research at University of Munich, and then I'm not exactly how this works in Munich, uh, Magdalena, but like there's this Ludwig Maximilian University. Is that completely separate from University of Munich or?
No, so I'm at two institutions at the LMU, which is a university, the Ludwig Maximilians University, and I'm chair there of physiological genomics. And the Institute of Stem Cell Research is not university. It's actually belonging to the Helmholtz Center Munich. This is a non-university basic
Okay.
funding or research organization. So it's a little bit like the Max Planck, so a
I see.
non-university organization. And I'm heading the Institute of Stem Cell Research there.
Yeah. Okay. So when I was in the 1990s, I remember that there was like a group in Lund, Sweden, and maybe some other places that were transplanting human embryonic stem cells into the brains of patients with Parkinson's disease and not really seeing any dramatic beneficial effect. And then a big breakthrough in trying to replace cells that are lost in whatever, Parkinson's, Alzheimer's, even stroke, I guess, or traumatic brain injury is Shinya Yamanaka's and his group in Japan developing a way to convert human skin fibroblasts into pluripotent stem cells. And then many investigators since then, including you working on this, have developed ways to then take those cells that are capable of forming any cell in the body or brain and directing them to a particular type of cell. Okay, so that's good, and that's been done in a dish. But then there's still another big problem. What do you do? You have to transplant. I guess the approach would be to transplant them into the brain, but that's can be problematic too because it's surgery and you're putting in cells and it's unclear. So you've been working on something that's really exciting and it's kind of space, it's almost science fiction in a way, but being able to convert non-neuronal cells, glial cells in the brain directly into neurons. And so that's, what I'll have you talk about today. This is really exciting research, and from my point of view. Um, Before you start going through all the details, where there's a lot of technical details and so on, and hopefully you and I can kind of try to make it explainable to people with kind of a basic science background, but may not be in this specific area. But before you get to that, can you talk about your background? Where did you go to college initially? How did you get interested in neuroscience? And then what led you to study stem cells and brain development, which you've worked on too?
Yeah, sure. Happy to. I studied biology and I was really always interested in biology already at school. I loved that. We also had a great teacher in that subject. And so I studied biology in Tübingen, here in southern Germany. And I fell in love with developmental biology relatively early on in a practical course at the Max Planck Institute. And ever since, I've been fascinated by how in development different cell types are generated, how stem cells are particularly plastic and can generate different types of cells. So in the nervous system, the neural stem cells, they can generate glial cells, but they can also generate neurons. And I've been fascinated since my studies and then my PhD, still in Tübingen, about the mechanisms, to understand more about the mechanisms, how these stem and progenitor cells actually generate neurons or generate glial cells. I followed this interest up in a postdoctoral work at the NIMR in London, UK, with Jack Price. And then when I came back, actually, the breakthrough discovery that led me into reprogramming also was that we found that the neural stem cells in the developing brain are actually glial cells. They are the so-called radial glia. And that was really a small revolution because everybody had thought that you have subsets of stem cells, so to say, that generate neurons, and you have others that generate glial cells, and then you have these boring guiding structures that are radial glia cells that would only serve for neurons, newborn neurons, to migrate along. And we could actually show that this was not the case. Now, we could show that when I started my own lab at the Max Planck Institute of Neurobiology, as it was called at that time, here in Martinsried, south of Munich, where I started my lab. And I'm still in Munich. I also met my husband here. So now I got tenure then and got appointed to these positions that were so confusing to you. Now, let me move on from the discovery that glial cells, radial glia cells in the developing brain, act as neural stem cells, because that then made me think, okay, these radial glia cells, literally, they don't exist anymore in an adult brain. Yeah. But of course, some relatives exist, namely so-called astrocytes, star-shaped glial cells. And indeed, these findings about the radial glia cells then triggered me to explore if maybe we could turn these astrocytes into neurons. Now, I was working anyway on neurogenesis, and I explored a transcription factor that we found in these radial glia cells during neurogenesis in development when it's up and running at its peak. And this transcription factor was and is PAX6. So I started then to put this transcription factor PAX6 that is important for neurogenesis during development, to put it into later glial cells, astrocytes that we took from a postnatal brain. We put in a dish first to see, can we really elicit neurons from these postnatal astrocytes that normally would not generate any neurons at all. And indeed we could, and as you mentioned, the induced pluripotent stem cells, we actually showed this direct reprogramming of glial cells into neurons in a dish, but even the first in vivo experiments in a mouse brain before the invention of the induced pluripotent stem cells.
And that was just with expressing PAX6.
First, we did it with expressing PAX6, absolutely. First in vitro and then in vivo. Afterwards, we found more potent transcription factors that give us beta neurons that then fire action potentials and are more mature. And much of this work was done with Benedikt Berninger, who now has his own lab and his own standing in the field of direct reprogramming. He's at King's College, UK. Exactly, and I can dwell on that in a second, but where we went from there and where we are at the moment with this approach, But let me briefly dwell on this timing there, because before the induced pluripotent stem cells, we also had a hard time because people just were very critical about reprogramming. Maybe these were not converted, and maybe you had 1 in a million stem cells left in your culture, and these proliferate and give you all these neurons. And of course, we did control experiments to show by genetic fate mapping that the cells really came from astrocytes. Right. But the induced pluripotent stem cell discovery, which of course showed the power of reprogramming, I don't want to compare our discovery with this, but it also really helped us a lot to get this accepted, that indeed cells can convert. And like you said, it sounds like science fiction, right? It's amazing. You have an mature astrocyte, and then you turn this in a neuron, and of course even more amazing, you have a skin cell from a person, whatever, 60 years old, and you turn it into a pluripotent stem cell. So the power of reprogramming has really been unleashed thanks to this discovery. And now, of course, I'll talk more about the brain, but I want to make a general statement here that turning fibroblasts into cardiomyocytes is also very far advanced, moving it towards therapeutics for heart disease and heart attack. So just a general praise of reprogramming, driving maybe the future approaches in medicine for diseases where we lose cells that can so far not be replaced, like neurons in the brain and cardiomyocytes in the heart.
So the conversion of astrocytes into neurons, it seems to be like a multi-step process in a sense that requires more than just one transcription factor. So maybe the PAX6 will cause the glial cell to become post-mitotic and maybe start to grow some processes, but additional differentiation steps have to occur to get the cells so it'll become electrically excitable or form connections. Is that?
Just for the sake of the argument, I say no. I anyway like to say no. It's one of my favorite. No. Indeed, we can do it with a single factor. For example, the proneural factor, the proneural transcription factor, neurogenin 2, or there is another one from this class, ASCL1, that has also been shown to be very potent in turning even fibroblasts into neurons, right? We can not only do it with glial cells, one can also do it with fibroblasts. So in principle, a very potent proneural neurogenic factor is sufficient to drive an astrocyte or even a fibroblast right through to an electrophysiological active neuron. So for the non-experts, neurons talk by firing action potential, by basically changing their charge very fast and become electrical active. And this is the gold standard for a neuron that is functionally active. And indeed, this can be achieved with a single transcription factor. And you can make this single transcription factor even more potent by, for example, changing its phosphorylation sites. We had a paper on that last year. So I would say, in principle, a single factor is sufficient. Nevertheless, when you go in vivo, and now we talk about, performing a brain injury in a preclinical model, like a traumatic brain injury, and then you use a viral vector to infect the reactive glial cells, typically proliferating reactive glial cells. with the viral vector, then either a control or with our famous neurogenic factor that I just mentioned, for example, neurogenin 2. And then you come back 1 week later, 2 weeks later, 3 weeks later. You do find neurons. You find even some that are also electrophysiologically active. But many cells or many young neurons also die again. So then we actually found that co-expressing, for example, BCL2 or a factor that helps the neuron survive, or a factor that helps the metabolic functioning of the neurons, then can really improve the neuronal survival, let's say, of these reprogrammed neurons. So therefore, you are right also that, combining factors, of course, helps, but the principal conversion can be achieved to a very differentiated neuron already using a single factor.
So these different transcription factors that you talked about, so presumably they're controlling the expression of a whole bunch of genes that when you express them, they turn theirs on. There's also transcriptional regulators that suppress production or expression of certain genes. Like one we played around with a little bit is called REST.
Yes,
And it
absolutely.
inhibits a lot of genes that are genes that encode proteins, neuronal specific proteins. So is there any value or in knocking down or reducing levels of these repressors that prevent?
Absolutely.
Yeah,
Yeah, absolutely. Very good point. And indeed, we showed some while ago that REST is an inhibitor of this conversion, and it particularly comes up when astrocytes mature a bit further. And we had a very simple in vitro model then still, where we had our reprogramming factors inducible. And when we just waited a bit longer with the induction, just about a week or so, and then induced these reprogramming factor, we got very poor conversion rate. And then we explored what's going on, and indeed REST was the culprit. REST came up even more and blocked much of the conversion, and when we could knock it down, we could improve that. Absolutely. In vivo in particular, we also found another inhibitor, if you want. This was the transcription factor OLIG2, which is known for its role in glial cells, and it's also known for the fact coming up in reactive glial cells. Classically, it's in oligodendrocytes and their progenitor. These are the glial cells that put the insulation on the axons, amongst many other things they do. And the astrocytes, the other class of glial cells that I was talking about, they typically upregulate this transcription factor after injury, at least some of them. And what we could then do, actually in the first in vivo study already, when we used PAC6, we could then use a dominant negative version of OLIG2. As you mentioned, these are repressors, and OLIG2 is also a repressor. So a dominant negative version of a repressor is basically the OLIG2 DNA binding domain fused to an activator domain like VP64 or so. So when we combine that with our neurogenic factor, then we got much better results. So you're perfectly right that we have repressors of neurogenic genes, and in certain circumstances, we can apparently override them with our proneural factors. But definitely they are there, and if we antagonize their function or knock them down, things work even better. Yes.
Okay, so in cell culture and in vivo, you can express or repress certain factors and get astrocytes to stop dividing, grow axons and dendrites, form synapses, become electrically excitable. Now, what about different types of neurons? So all neurons aren't created equal. There's
Yes,
glutamatergic, GABAergic, dopaminergic,
yes.
etc.
Yes. So let me correct one small
Okay.
aspect which you mentioned already twice, I think, namely that these reprogramming factors would make the astrocytes divide. This is actually not the case.
No, stop divide. Oh, they don't make them
No,
stop.
no, no, they make them stop
Yeah,
divide.
isn't that what I
Exactly.
said? Okay.
Okay, I understood the opposite, but
Okay.
anyway, it's always good to clarify.
Yeah.
Absolutely. They make them stop divide. If we in fact dividing one, we can also convert non-dividing
Yeah.
one, but generally it works better for the dividing ones. Exactly. So they make them post-mitotic, like in development, right? The
Yeah.
stem cells are proliferating, but these neurogenic factors drive cells then towards neurons and they are post-mitotic. I'm emphasizing this because a very often asked question is, of course, who is there danger for tumor formation
Yeah.
also? And
Yeah.
they actually act the other way. So
Mhm.
this would not be yeah, this would not be at first glance such a danger. But now to your question. Indeed, one can make different subtypes. For example, using the two factors that I already mentioned, the neurogenin 2 in development in the forebrain, for example. Neurogenin 2 is involved in making excitatory glutamatergic neurons. If you look in the cortex, pyramidal neurons, glutamatergic projection neurons. Conversely, the other factor I had mentioned, ASCL1, or formerly known as MESH1, but these are all details, names don't matter here, is expressed in the ventral telencephalon, if we stay with the forebrain, and there it makes GABAergic neurons. So already many years ago, when we had our murine postnatal astrocyte cultures, we tried both of these factors, or each of these factors, I should rather say, and indeed we found that we generate different types of neurons. Using ASCL1, we get GABAergic neurons, and using neurogenin 2, we get glutamatergic excitatory versus the inhibitory neurons. So that shows you that indeed these developmental factors activate, probably similar genes as they do in development and drive then these cells towards a certain neuronal subtype. Having said so, it's not always as simple, and here with just using one factor, and here your point from before is very appropriate, because, for example, to make dopaminergic neurons, different labs are using at the moment, I believe, 5 different transcription factors. Also, to make, for example, cortical projection neurons that would project to different targets is still like, we call them upper layer neurons that project to the other hemisphere, for example, or everybody has probably heard about corticospinal neurons that go down to the spinal cord and are very important in the motor system. This task to make these specific neurons, I would argue, has not yet been achieved, and the community is actively working on finding the good combination. Now, and therefore your point is absolutely correct. While we can generate a neuron that fires action potential, sends out axons, connects to other neurons, receives synapses. We have recorded from these neurons. This all works with a single factor, at least for many of them. But to make real subtypes, we need combinatorial factors, and that's the state at the moment that we are at, I would say. Yes.
And then there's a couple other things you've worked on in terms of this differentiation process. I guess one is microRNAs, which uh, there's been a lot of interest in, and these are, well, they're small RNAs that interact with messenger RNAs, right? And inhibit.
Now also, yes,
Yeah.
yes. Yes, so there are protocols, mostly, I mean, particularly beautiful and developed by Andy Yo, for example, in the US, using two microRNAs. He gets, or his lab gets, a very beautiful conversion also of fibroblasts into neurons using two particularly potent microRNAs that are also in a regulatory loop, let's say, which is really important here. And then he gets, let's say, a pan-neuronal identity, but then to make dopaminergic neurons or particular other neurons, you have to come with some other factors to give them a further identity. But it's very good that you mentioned these microRNAs. They can amazingly also do the job, but you need a bit more then.
Yeah, and this gets really in vivo, it gets complicated because, for example, if you use to get a gene in or a microRNA, I guess you use, say, adeno-associated virus.
No, I said
It,
virus so far, and I would not at the moment. Adenovirus is a loaded term because
ah,
the adenovirus created some artifacts. So,
ah. Even AAV.
well, especially AAV created some artifacts
Huh.
because, so let me start again.
Yep.
When we did the first in vivo reprogramming in the mouse brain, we used a Maloney-based retroviral vector
Okay.
that
Ah.
can integrate only in
Yeah,
dividing
that makes sense.
cells
Yeah.
its RNA. So therefore, you are sure not to target any post-mitotic neurons, right? Because neurons don't divide. And so therefore, of course, you do your controls, but therefore you are sure that then your neurons that you see, they really come from astrocytes. And of course, we did further controls for that. Now, AAVs are, of course, less immunogenic, and we and others have then started to use them. But how do you direct your AAVs to the
Right.
astrocytes only, right?
Right.
And there really an unexpected artifact occurred because you use your AAV with your control, GFP, whatever, beautiful, only astrocytes, only glia. You use your AAV with, for example, a GFAP promoter region, driving your neurogenin 2 then. And of course, in the control, you also use this promoter region. I just forgot to say. So then you use again this GFAP promoter element, and you drive your neurogenin 2, for example. And then slowly you see more and more neurons appearing.
Okay.
Sounds like perfect reprogramming, but wasn't. Because in fact, genetic fate mapping then showed that actually it was an artifactual expression of the reporter in endogenous neurons.
Huh.
The neurogenin
Ah,
2,
I
this
see.
proneural factor, activates your marker, your GFP, your marker gene. And then neurons appear and you think, great, but these are not reprogrammed neurons. And actually one could see it because they are perfect neurons,
Ah,
right? Right from the start. Normally the neurons, especially in an injured brain, they look a bit scrambled. They don't look perfect,
right.
really.
Yeah.
So therefore, you are perfectly right that, let's say, if we envisage a therapeutic setting, most likely one would go and use less immunogenic viruses. One could also these days think a lot about RNA delivery. So more and more tools are developed with that. But certainly, in an ideal setting, one would have a viral vector or an RNA packaged that one can inject, ideally IV, or, you know, one doesn't have to operate the brain. And this viral vector would then go into the brain and target specifically astrocytes. And I saw from your face that you think it's science fiction, but it isn't science fiction. There are indeed AAV vectors that you can inject systemically and that go to the brain. I mean, in an injury, the blood-brain barrier is anyhow open, but there are viral vectors that can go via the blood-brain barrier, and they have a tropism for specific cell types, for example, astrocytes or other glial cells. And on top of it, one could say that expressing these neurogenic factors, maybe even in the neurons, actually helps the neurons to survive after injury. These are new data that we haven't published yet, that we have in the lab. So, you know, you're perfectly right for the therapeutic setting. But in the preclinical experiments, this has steered up quite a bit, because there were famous Nature and Cell papers claiming that, you know, you just need this one factor that you would actually knock down, and then in the retina you get beautiful ganglion cells, in the brain you get dopaminergic neurons. And so of course you did, because these were all the endogenous cells. So as always, careful with interpretation of results, always do controls. We wrote an article about it, a review article in Neuron calling for gold standard controls.
Yeah, I'll put that. I will make a link to that article because it's really good.
Yeah,
Yeah.
cool, cool.
All right. So you've done some work with traumatic brain injury model, right? And
Yeah.
and what's the current state of that? What have you accomplished so far?
Yeah, so I would say we showed that it works in vivo.
By works, do you mean do they get recovery of function?
Yeah, recovery in function is a special question.
Yeah, that's kind of the ultimate question, right?
I mean, let's put like this. In that regard, we have already many treatments, because you can transplant, for example, mesenchymal stem cells, and you get improvement of function. But this has nothing to do with making new neurons and really replacing neurons, right? But of course, it can help the patients. So we are working on really replacing lost neurons that then integrate into a neural network. Exactly. And so far, what we and others have shown is that you get functional neurons, they're electrophysiologically active, they also receive synapses. Functional output hasn't been shown yet, but it's also very difficult even for transplanted neurons. And we are now making the next step before characterizing everything to death, so to say, in a preclinical, I mean, with mouse cells. We made the next step now to go to human cells. So we differentiate human iPS cells into glial cells. We convert them into neurons, like with a mouse, first in a dish. And now we are exploring them. So we transplant the glial cells into the injury model, and then we convert them there into neurons. And so in my view, that is one of the possible roads one can take to probe that human reprogrammed neurons can integrate into a network, that they can, you know, be functional using calcium imaging and these things. So at least from my perspective, this is where we are now. notwithstanding what you mentioned before, that one needs to work more on the neuronal subtypes. So we are not working with Parkinson disease, but you mentioned the Lund experts, for example, Molin Palmer is very active there, and she is working with transplantation, but also with reprogramming. And then one needs to do that in the Parkinson models, for example.
It seems like for the, I guess you'd call it in situ direct programming, it seems that that for functional outcome measurement, you really need to have an injury that only damages a small number of neurons, but that causes a clear behavioral effect. A lot of people have done work with things like certain neurotoxins like canic acid that selectively kills, say, CA3 or CA1
Yeah,
pyramidal
yeah,
neurons, or 3NP that selectively damages striatal medium spiny neurons in a Huntington's disease model.
exactly.
Um, so, I mean, you can only do so much, but have you tried any of those models?
We haven't, so we stay in the neocortex with our injury. We do a lot in stroke now, so
Ah,
stroke would
yeah,
be the application that we would go for. But a former postdoc from my lab, Christoph Heinrich, who works in Lyon now, he has shown in an epilepsy model
yeah.
that reprogramming into inhibitory neurons,
Oh,
GABAergic neurons, can indeed improve
geez.
function. So
Yeah,
basically reduce the epileptic seizures
yeah.
in this model. So in that regard, that has been shown. Yeah.
Well, you may know my lab did a lot of work on kind of environmental factors like
Yeah.
lifestyle factors like
Yes.
intermittent fasting or physical exercise.
Yes.
And it seems for stroke, for example, recovery from stroke,
Yes.
like the patients do physical therapy and it does help.
It helps a lot. Yeah.
So, maybe eventually there'd be a combination of an intervention to cause direct programming and get some neurons and new circuits forming in combination with some environmental factors that are known to,
Absolutely.
for example, to upregulate neurotrophic factors or
Absolutely.
so.
Yeah.
What about neurotrophic factors? The injury itself will induce That's how, for example, BDNF was really discovered in a seizure model. Uh, they found that when they give canic acid, they get dramatic increase in BDNF mRNA
Yeah.
and
Yeah,
hippocampal neurons.
absolutely. Yes, absolutely. So generally, I couldn't agree more. And in fact, we have also colleagues here who work on even the formation of detour circuitry and exploiting the plasticity of neurons after injury, and including rehabilitation, treatment. So we also team up with them, and we also do some experiments with enriched environment or running
Ah,
wheel
yeah.
of these mice. It's very important to get the timing right.
Ah,
And we are still actively
yeah.
exploring that. Do you do it early, or do you do it actually more at a later stage when the glial cells have already been converted into neurons? And then, so yeah, there is a lot to play around with, but you are absolutely spot on. Surely it will be some combinatorial
Yeah.
treatments, or like I mentioned, to keep the cells alive, especially throughout a critical step in this conversion. So many things that may factor in there. Yeah, absolutely.
Okay, then another thing that I think you worked on a little bit, I know others have, and there's been some, I think, papers retracted and stuff based on it, but using like small molecules to to affect the phenotype of the cells, like Notch inhibit chemicals that inhibit Notch, or that activate certain signaling pathways like cyclic AMP, or
Yeah,
maybe even calcium. Calcium's such an important uh, second messenger for all sorts of different
absolutely.
pathways. What's
But
going
it's
on there?
difficult to, stimulate it in such a way that it will drive or foster the conversion of a neuron, because it's important in astrocytes and it's important in neurons.
Yeah.
Yeah. But definitely small molecules are important. And for example, the standard protocol in vitro actually involves two, at least two, I can think of more, two of these factors. Phoscolin is often added to get these neurons more mature. Dosomorphin is often added to, inhibit the glial fade a bit from these. And, you can add much more. And as you are referring to, the lab of Hongkui, for example, is famous for using only small molecules for reprogramming, and had, after all, the first beta cells derived
Oh,
from such an approach,
uh,
transplanted into patients. So certainly,
yeah,
a lot of things are
the pan,
possible.
we're you're working on the hardest organ to, right? So you mentioned the heart, right? Where if they have a heart attack, the idea is to replace the cardiac myocytes that have been lost, or if you have diabetes, I guess type 1 diabetes, and have lost the beta cells in the pancreas, it's just a small organ, not so many complexities.
Yeah, absolutely. The brain is the hardest in that sense. And like we were discussing before, it's super important that the neurons integrate adequately, that they don't do any harm then, right? Maybe we make neurons, but if they don't connect properly, maybe they make some aberrant circuitry. This is what we need to study now, but for example, for epilepsy and the interneurons, it can already help. For dopaminergic neurons, we also know, even so they are transplanted ectopically, meaning in another brain region than they normally reside in, namely the striatum, the target region, so to say, of these
Huh,
dopaminergic neurons that
huh.
are dying in Parkinson disease patients. Nevertheless, they can help. So I think,
Oh,
there
so
are
the
applications.
substantia nigra is dispensable. We don't really need it. No,
Naja,
I'm making a joke. But you're saying they put the dopaminergic neurons right in the target, the striatum that they
that's how
normally.
they do.
Yeah.
That's how they do it. That's how
Yeah.
they started it in Lund, and now several clinical trials using iPSC derived or human embryonic stem cell derived dopaminergic neurons are again ongoing. So these replacement therapies are revived from the transplantation side, but also from the reprogramming side. Yes.
Now, astrocytes. So when there's a brain injury, the astrocytes can change their phenotypes. They become like reactive or so. But another type of glial cell from the standpoint of injury is maybe as interesting, the microglia, because they're kind of all over the brain. They kind of move around, but when there's an injury, they like congregate where the injury is. So it seems maybe if you can convert the microglia to neurons,
Mhm.
there might be some advantage there. What about that?
Absolutely, absolutely. So this brings us to the interesting point. If one uses the cells that are easiest to convert, or if one uses the cells that are maybe neurotoxic, for example.
Well,
There's
yeah,
also subtypes of
yeah.
microglia that are neurotoxic. So if you want to catch two birds with one stone or so, or the cells that are most concentrated at the lesion, as you mentioned, absolutely. Now for the microglia, there are again some contested papers claiming that they could convert microglia into neurons. Then it has been challenged, let's say. Then this lab has provided further evidence. So we haven't done that, but let me summarize. It seems not so super easy maybe to convert these microglia, but certainly it can be done. One can also reprogram fibroblasts into neurons. One may need more factors. And this is certainly also a field that's being worked on. Exactly. We stay with the astrocytes because we were also very excited recently. We had a Nature Medicine paper in 2023 where we discovered that also in the human brain there are proliferating astrocytes. And as we think they are, also a little bit de-differentiated, more partially more like radial glia cells, we really think they are the easiest to convert. So we think here we have a good cell type that we could maybe target to achieve that. But absolutely, other glial cells may have an advantage there as well. Yeah.
Right. And then there's even another one we didn't mention, the oligodendrocyte progenitor cells that give. Right.
Absolutely.
Now, as far as myelination, right? So neurons with long axons often have, they're wrapped by the oligodendrocytes, the myelin that increases the conduction velocity. When you reprogram the astrocytes into neurons in vivo,
Do they myelinate?
can they become myelinated?
Yeah, you have very good spot-on questions because we just discovered that even the transplanted neurons are actually not getting myelinated. So we have 30 years of transplantation research, and people didn't look systematically. And indeed, the student in my lab just had the project to look if the myelination is correct, internode length, is it correct? But then she looked for myelination and oops, there were hardly any axons myelinated. So now she has spent a whole PhD in trying to overcome this for the transplanted neurons and has found something looks like promising, still ongoing. But yes, absolutely. So again, for dopaminergic neurons, right, it's not needed. They are not myelinated. Also, you put them on the target site, so you don't need it. If we want to replace corticospinal neurons, we certainly need to instruct myelination, both for transplantation approaches or reprogramming approaches. And yes, we are working on it.
Yeah, it gets so, again, we come back to the complexity of the brain and all the little hurdles that have to be crossed and
Absolutely.
how you're going to approach it technically and from just even from, can you do it? The first question in experimental systems, and then two, how would you translate that to humans? What is going on with humans? So you mentioned in the heart, there's already clinical trials. Do you have to quit right at on the hour? How much?
Yeah, but we still have, we
I have
still
a
have
few
some
minutes.
time. Yeah.
So what's going on in the clinic? So you mentioned there's clinical trials in heart attack patients or with
I
what?
don't think it's yet in the state of clinical trials, but there's already companies working on it. So it's certainly advancing
I see.
towards therapy. And of course, in a way, you know, it's indirect, but nevertheless, half of the clinical trials that are at the moment ongoing with pluripotent stem cell derivatives, half of them are with induced pluripotent stem cells. So after all, they are reprogrammed,
Yeah,
right? And
yeah,
there's this idea of autologous
yeah, yeah,
collecting patient cells from the skin, turning them into iPSCs, differentiating these
yeah,
into dopaminergic neurons, for example, and then putting them back in the brain of the patient her himself, avoiding all the immune suppression and so forth. So reprogrammed cells in that sense are already used in clinical trials, but so far only in that sense of, we can call it indirect reprogramming. I would say really, like you mentioned, it's important to mention to the public that the direct reprogramming approach is not yet
right.
close to the clinic. We are still working on basic aspects like neuronal subtype integration and so forth, but there are routes that will be faster going towards clinical trials, maybe the interneurons and other things. And it's important to mention that also the transplantation is just going back to the clinic for the brain repair. There was a big gap since the first
Yeah,
attempts
yeah,
because one was using fetal tissue mostly, and now using pluripotent stem cells. New trials have been instigated, and while we are now, you know, expressing some words of caution of where things are with this in regard to clinical therapy, it's really encouraging to see that now stem cell mediated trials are really on the increase again. So it's good times. There is positive notes, but of course, still a lot has to be done. So Alison Abbott wrote a very nice short article in Nature, in the first issue of Nature this year, where she was overviewing the stem cell, clinical trials that are now ongoing using stem cell derived cells. So worthwhile reading. So positive note on hopefully making real progress in replacing cells that are lost due to disease by transplantation or by reprogramming. But for the brain, indeed, still some work is needed.
So maybe some of your graduate students and postdocs that are going through your lab will, be involved in that 20
Yes,
years,
like I
20,
mentioned
20
some
years from
already.
now or
Yeah.
something.
Yeah. Well, we are, of course, also trying to continue to help push it forward using the human cells and transplanting the human glial
Yeah.
cells and turning them into neurons. So we help pushing this forward, but we certainly need many hands to help.
Okay, you keep working on that. I'll keep working on risk reduction and
Perfect.
prevention side of it.
Yes, very good.
Oh, that's yeah, because some of these disorders, a major risk factor is obesity, insulin resistance, people don't exercise and so on.
Sure.
As they get older, they're more vulnerable to stroke. They have worse outcomes if they have traumatic brain
Absolutely.
injury. We think Alzheimer's, your lifestyle factors are also important. So, yeah,
Absolutely. Prevention is always better than repair. I think this is something we can say for every organ we have, right? We better prevent damage than having to repair it. But we are working on the cases where the damage has been done already.
yeah.
But generally, prevention much better.
Okay, thanks, Magdalena. I enjoyed talking to you. It's good to see.
Likewise,
It's good
likewise.
to see. I'm semi-retired, so I'm kind of having fun doing this and some other things.
Cool. I look forward to that stage then.
Yeah. Okay,
Okay,
yeah.
it was really fun
If you
talking.
want any advice, let me know about that.
I will. I will. Okay. Great to talk to you,
Yeah.
and let's stay in touch. Absolutely.
Okay,
Take
goodbye.
care. Bye.